Good afternoon, everyone. Thank you very much for this kind introduction, and thank you to the organizers for inviting me to this meeting. It is a pleasure to be here. It's my first time visiting St. Louis and seeing the arch in person. I did not realize how large it is. I thought it was slightly smaller, at least, based on the photos I've seen. Without further ado, I'd like to share with you today some insights on P53 alterations in myelodysplastic neoplasms and acute myeloid leukemia. P53, being an important gatekeeper of our genome and other important cellular functions, is not surprisingly an important regulator of hematopoietic stem cell functions. Of course, under physiologic conditions, but also within the framework of myeloid neoplasia. So P53 expression is tightly regulated within the hematopoietic stem cell compartment. The importance of P53 within that compartment is manifest through studies that have shown that pressure to hyperactivate P53 in conditions such as Fanconi anemia and other type of inherited predisposition syndromes plays an important role in bone marrow failure. Similarly, P53 alterations have been demonstrated in clonal hematopoiesis as one avenue through which leukemic or pre-leukemic hematopoietic stem cells acquire a survival advantage and thus create a situation where a clone in the bone marrow is able to proliferate in a somewhat uncontrollable manner. And of course, as we will see in the next portion of this presentation, with an emphasis on today's topic, is P53 playing a role in myeloid neoplasia and the progression of neoplastic myeloid cells through the MDS-AML continuum. A few words about P53 at the gene level, and then in the next slide at the protein level. Just to level set, alterations of P53 fall into two main categories. One category is the loss of one of the P53 alleles through copy number deletion. And the other category is P53 variants that result in a mutant protein which has a compromised function across all aspects of P53 biology. It is important to remember that through evolutionary forces, there is protective redundancy that allows cells actually to lose a single copy of the P53 gene locus and yet remain viable and alive. Loss of both alleles, of course, is incompatible with life. It is on that basis that it might not be surprising to know that 60% or so of cancers in humans have P53 missense mutations plus concomitant deletion of the other allele. So it's not just the presence of a P53 mutation that answers the question of whether there is an alteration in P53 biology within a certain cancer, but it's really a combination of copy number loss plus alterations at the functional level. So without going into too much maybe biological details today, there is some protective element for the presence of an intact P53 allele within the context of neoplasia. In addition, at the genetic level, one has to keep in mind that the implications of P53 mutations depend on the type of mutation that we're talking about, on the site of mutation within the various functional domains of the P53 protein, and the presence and whether additional mutations, so whether we're talking about a single mutation in the P53 gene or multiple mutations, that can determine the functional implication within the realm of cancer. At the protein level, we have known for many years that mutant or variants that result in alteration of P53 biology result in accumulation of the p53 protein within the nucleus of cells. This accumulation is influenced by a number of factors. It's not a simple mechanistic, there's no simple mechanistic explanation for it, but rather a conglomeration of factors that include the steady state of ubiquitinization of the p53 proteins, association of the protein with molecular chaperones, protein folding, messenger RNA levels, and so on and so forth. In these immunohistochemistry stains that you see on this slide, you see on the left-hand side a normal pattern of p53 expression in a bone marrow core biopsy. You see that some cells express p53. That's seen with the brown signal within some of the nuclei. The brown signal within that normal bone marrow range from very mild to strong, as you can see in a few cells. Let me see if I can... I'm probably going to skip the pointer. I'll leave it to you to hunt for those cells with high p53 expression. Well, on the right-hand side of the slide is mutant p53 accumulation. And there you see a uniform high-level staining pattern for p53 within the nuclei of cells in this bone marrow with acute myeloid leukemia. Again, demonstrating accumulation of the protein within these nuclei. Now, we have long known through many studies that have emerged over the past decade that myelodysplastic neoplasms can harbor p53 mutations, and typically such mutations tend to associate with higher risk myelodysplastic neoplasms. People have demonstrated that mutant p53 in MDS with isolated deletion of 5q, which I'm not showing here, actually imparts some level of resistance to treatment approaches that otherwise work in these patients. We demonstrated a few years ago that 47% of myelodysplastic neoplasm cases with significant fibrosis, that's significant reticular fibrosis in the bone marrow, have p53 mutation as a hallmark feature. These typically are aggressive, high-risk MDS types. Within that study, we were also able to demonstrate that the mutant p53 protein expression is not restricted to the stem cell compartment, which in this image shows, is highlighted by some red staining for CD34. So the red stain around some of these cells with p53 high expression are blasts by virtue of CD34 expression. But you can also see that p53, mutant p53 is expressed in more mature elements within this MDS, cells that are not CD34 positive. And so the mutation, the clone that harbors the mutation, carries that mutation forward throughout its differentiation line. Other groups have demonstrated that beyond just the mere presence of a p53 mutation, and this will be a theme that we will come back to over and over again today, the genomic context in which these mutations arise is also important at a clinical level. So the allelic frequency, as well as the copy number status, impart an important element to understanding the role of p53 in MDS. This is a paper by Dr. Garcia Manero's group from MD Anderson, led by Guillermo Montalban Bravo, again showing, and I point your attention to the bottom left corner of this slide, showing that p53 mutation, complex karyotype, and copy number loss at the p53 locus are not really always codependent, right? So there is an element of independence to these events, and it is the overlap, the presence of 2 or 3 of these conditions within a single case that imparts the most biological significance. I would say that notion got cemented a few years afterwards in this landmark paper from the Memorial Sloan Kettering group, in which several thousand cases of MDS were surveyed to understand the impact of copy number status in the context of MDS and P53 mutations. And it was clear based on this study, which now is the basis upon which, as you will see, the classification of MDS has been reformulated to say that it is the multi-hit P53 state and not merely the presence of a P53 mutation that determines or imparts, again, as we said earlier, a unique genetic signature to MDS. So that was the information that was available against which we looked at data in the literature to inform us on what to do with regards to P53 and myelodysplastic neoplasms. It is not my purpose today to go over the WHO classification. I would like to draw your attention, though, to the category of myelodysplastic neoplasms here. I'm putting it in the context of other important related myeloid neoplasms in the 5th edition of the WHO classification. You will notice that for the very first time, we have separated MDS into 3 families. One is MDS with defining genetic abnormalities. The second family is MDS that's morphologically defined. And the third is MDS of childhood. Under MDS with defining genetic abnormalities, you will see that the third entity there, the third MDS type, is MDS with biallelic P53 inactivation. And it is really the data that I shared with you that, in addition to some more that I'm not sharing today, that really gave us the foundational elements to make that change. Taking a step back and thinking about myeloid neoplasia more broadly, the WHO classification has created, I would say, cleaner and clearer boundaries that help us draw lines between, of course, benign conditions that cause cytopenias, clonal cytopenias such as clonal hematopoiesis and clonal cytopenia of undetermined significance, MDS and AML. For the very first time, the WHO now recognizes clonal hematopoiesis as a pre-neoplastic condition and has created a working definition around that disease state. Drawing a line between CCUS and MDS is an emphasis on MDS being defined on the basis of morphologic dysplasia plus cytopenia. And it's within that context that now we have this new entity of MDS with biallelic P53 inactivation. And I am taking the essential and desirable diagnostic criteria from the WHO classification and highlighting them here. So to make this diagnosis, we need cytopenia, dysplasia, and blasts being under 20% in the peripheral blood and bone marrow. We also need to detect one or more P53 mutations to make it to this diagnosis. Now, in the presence of a single P53 mutation only, direct or indirect evidence of P53 copy number loss or copy neutral LOH would be required to make this diagnostic, to make this diagnosis. It is desirable to have a complex karyotype, but not essential. Now, how does this translate into day-to-day kind of impact? This is a paper that was published by the MLL group in Munich, Germany a few months ago, showing that, and I point your attention, it's a busy, sorry. I point your attention to this entity here, MDS with biallelic P53 alterations. And if you walk it back to where cases were prior to the institution of this entity, you notice that most patients come from MDS with excess blasts. Some of them come with excess blasts too. Some come from MDS EB1, But interestingly, some patients actually would have been called previously MDS with 5q deletion, MDS with multilineage dysplasia and ring sideroblasts, and so on and so forth, kind of feeding into this new entity up here. Now, what about P53 in AML? So I think we've, in a way, put P53 to bed in MDS. I'm not saying the story is ever closed in any classification scheme, but we did have enough data at the time the scheme was being developed by multidisciplinary experts, many of whom are in this room, to be able to kind of have a bit more firm footing in MDS. In AML, the story is a bit different. You see that AML in the WHO classification is split into two groups now, AML with defining genetic abnormalities and AML that's defined by differentiation. There is no AML with P53 mutation in the WHO classification, and I hope to spend the next 5 to 7 minutes at least providing some data that argues that P53 mutation per se as a defining molecular alteration of a type of AML is probably going to be masking a lot of important biological depth that we need to retain in our classification scheme. A few years ago, we published this paper showing that it is P53 copy number and protein expression status that together can inform basically decision points and prognostication and classification frameworks for AML with P53 mutation. In this study, we demonstrated that P53 mutations in AML actually span multiple domains of the gene. They are concentrated in the DNA binding domain, which is highlighted in orange in the upper portion of the upper left portion of this diagram. But that there are also multiple types, of course, not surprisingly, multiple types of P53 mutations. And that when we study these mutations in the context of copy number alterations, the picture gets a bit more complicated than merely talking about mutation status. We also showed that in addition to finding mutant P53 protein expression manifests itself as overexpression of the P53 protein by standard immunohistochemistry, we noticed that a sizable subset of AML cases can have an abnormal P53 protein expression pattern by IHC that's characterized by loss of the protein, by loss of signal. The protein is arguably still there, but it is not detected with the standard antibody that most people use in clinical practice. And last but not least, we showed that prognosis can actually be sum totaled on the basis of IHC and the protein expression status, and sometimes could provide a measure of impact, of biologic impact of P53 alterations in AML that transcends mere mutation status. What we were also able to demonstrate was that splice site mutations are not uncommon in P53 mutant AML. This could be detected in up to 5% of cases. These mutations are distributed around the DNA binding domain and can either coexist, can either exist separately, independently, alone, or coexist with other mutation types. We found one case, and this is a cohort of over 500 patients. We had two cases, where we had two splice site mutations coexisting in a single patient. And about a third of these patients could have actually the truncating pattern of P53 expression with loss of signal by IHC. This busy slide also illustrates the copy number status of P53 in the context of P53 mutations in AML. Without going into much detail here, of course, these slides will be available. It is important to note that the type of the potency of P53 mutations with or without copy number alterations is different. But looking at copy number loss by itself does not really independently carry a prognostic implication. And speaking of prognosis, this and the following slide are, again, busy slides to illustrate that when we look at when we do univariate analysis and ask about the granular components of P53 biology in AML and how they correlate with overall survival, and this is in patients who are previously untreated for AML. You notice that, and I'm going to draw your attention here to the variant allelic frequency, where a cutoff of 40% is associated with a worse hazard ratio, i.e. worse prognosis within this broader group, but that having P53 mutation in the context of certain favorable cytogenetic risk groups might not carry the same implication. Again, illustrating that mutation alone is not the entire story, but it's rather the multiplicity of events that associate with it. Similar findings for correlations with leukemia-free survival. Same idea, different metric. And when we looked at when we did multivariate analysis, interestingly, again, the VAF kind of emerged as an independent prognostic factor alongside mutation status. Again, suggesting that clones that harbor one or more P53 alterations, as reflected in the VAF, have different survival advantages depending on a constellation of findings related to this condition. This is perhaps an integration then of what we know about P53 in MDS and what we know about P53 in AML. Really kind of maybe can come together into a schematic that was just published about a month ago by Claudia Haferlack and her team that really illustrates that low-risk MDS going through MDS with increased blasts, more increased blasts, all the way to AML, is associated with increased rate of P53 double hit alterations. Single hit alterations actually kind of go down because by themselves such alterations do not correlate with a huge survival advantage all the time. And likewise, overall survival follows a similar trend within this group. And so as we look at the continuum of MDS to AML, again, we need to focus on the aggregation of mutation status, copy number status, as in the right context of blast counts, as the perhaps ultimate indicator that we're dealing with an entity that's dominated by P53 alteration from a taxonomy and classification perspective. Before ending, I think I'd be remiss in not pointing out that in certain types of AML, where we know that P53 alterations, oftentimes biallelic and multi-hit, are part of the overall picture, and this is true in pure erythroid leukemia, and this is a nice example of pure erythroid leukemia or acute erythroid leukemia in the new WHO classification. Even within this context, knowing that P53 exerts an important defining overall biologic framework, there is still room to make discoveries that are really impactful, both at a biological level and at a clinical and therapeutic vulnerability levels. This is a study that appeared recently. It's by Dr. Ogawa's group from Japan, showing that amplification of the erythropoietin receptor and JAK2 define a unique subtype of acute erythroid leukemia. Again, all of which pretty much have one or more p53 alterations. And that these EPOR and JAK2 gene amplifications actually seem to correlate, at least in their study, with sensitivity to tyrosine kinase inhibition, such as with ruxolitinib. So I think the story in AML is much, much more complex. And really, we should take it as cautiously as we possibly can, so that we're cognizant that the mutational and copy number alterations in MDS and AML are indeed complex, and that in MDS, biallelic or multi-hit alterations do define what seems to be a unique type, but that in AML, defining it on the basis only of mutant p53 would mask significant genetic variability and potential biologic vulnerabilities. And last but not least, and this is certainly an area that needs more studies, there are splice site mutations that involve the p53 gene, and that they appear to be impactful, and they require more studies because it is not clear that they're always detected in every mutation sequencing assay. With this, I end, and I thank you for your attention. Happy to take questions.
Thank you so much for that wonderful presentation. So the floor is open now for questions for the audience here and the participants on the virtual platform. I don't see the questions here. Am I supposed to see them? Okay. Celeste?
Hi. Thank you for the wonderful talk. I would like you to speak a little bit more about the intricacies of reading IHC for TP53, because I hear from my heme path colleagues that it's always a fun one to do.
Yeah, no, it certainly takes some getting used to. What we were able to demonstrate in the blood paper, and I didn't go into those details of the paper, was one, we were able to confirm that it's not just any staining for p53 that counts. It should be the intense, what we call 3+ out of 3 intensity that is biologically meaningful. In our study, the cutoff, the calculated cutoff based on area under the curve calculations using multiple methods that balance sensitivity and specificity turned out to be 7.2% for 3+ nuclei staining for p53. Of course, it's hard to say, what is 7.2%. From a practical standpoint, I typically use, basically, when you have a wild type pattern, it's oftentimes easy to detect. So saying that this is not p53 mutated is very important. And so that, I believe, with some exposure, ends up being a fairly easy thing. Most cases that have p53 mutation that's easily detectable by IHC often have a lot of staining. They're not really in contention. Whenever I'm between 5 and 10%, I hesitate. I don't really make a commitment based on IHC. And would defer to mutation analysis.
And in those cases, you're thinking that is the gain of function TP53s, essentially, that are doing that, right?
It could be that. So to throw another caveat, so that could be one thing, but another caveat is that FLT3 mutation actually, in the context of wild-type p53, can lead to a lot of upregulation of p53 expression. We've seen that with other types of activating mutations. So there's still more to be determined. I have found that using image-assisted evaluation of P53 IHC can help because the human eye is not always able to distinguish between 2+ and 3+ staining. But if you train and clinically validate an image analysis algorithm, it can do that job pretty well, I would say. So it's something that we need to gain even more experience with. It is a helpful tool, but it does take some getting used to.
Yeah, thank you.
Pleasure.
Hi, great talk. I just have a quick question as well. Did you see any correlation between the biallelic mutations and other gene abnormalities that justify the shift in the overall survival?
Just to be clear, a correlation between biallelic P53 and what else?
And other genetic aberrations that might drive the tumor, basically.
Great question. Interestingly, and again, that is also in the paper, I took the figure out, the mutational landscape of P53 mutant AML is actually quiescent. There aren't too many mutations in the background. The most frequent of those end up being RUNX1 mutation, which of course is another bad prognostic player in that context. But otherwise, the landscape is fairly quiet, so we couldn't find any correlation that would explain by itself any prognostic impact.
Interesting, thank you.
So the questions just disappeared. I'm trying to connect with my computer to see if I can pull them up, but it's not working. So I believe there was a question regarding the utility of, for TP53 copy number alterations, the utility of FISH and chromosome microarray. I think that's the question.
Absolutely. So I would say in day-to-day practice, for most of us, at least I can go back to when we were doing the study, we were looking at what the workflow was at MD Anderson when I was there. That was when we did the study. All patients with AML, all patients who are suspected to have AML would get mutation profiling, would get FISH, and would get ACGH. FISH was not always directed initially at looking for copy number loss in P53. I think with subsequent identification that could be an important player, that ended up being an additional piece of data. We were able to do FISH also on some of the cases that lacked that information in the clinical record. So I would say if the institution does ACGH or if your lab does ACGH, that could be an important tool that raises concern about copy number status at the P53 locus. If not, then maybe an algorithm could be that detecting a single mutation in P53 could, I think, justifiably be considered a trigger to do ACGH or P53, looking at 17P at the P53 locus specifically.
Thank you. So we will take a break now, and we will be back here at 4:15 for a Platinum Vendor Showcase by SOPHiA GENETICS.